Hi there guys, and welcome to video number 3 for year 12 A-level biology. Just to do these lessons obviously whilst people aren't at school, for either my students or anyone else that wants to use them, just to keep their knowledge up. If you're in year 12, it's a revision. If you're going into year 12, if you've just done your GCSEs in year 11, then this will hopefully prepare you for A-level biology and the standard needed. So the questions that you could do, so if you watched the previous videos, some extra questions, extra activities, see what you could do. You can pause them if you want to do them. If not, we'll move straight on. So today what we're going to look at is energy storage polysaccharides. Now, energy storage polysaccharides are polysaccharides which are chains of sugar molecules. And they're used in either plants or animals as energy storage mechanisms. And they mainly use alpha glucose. If you remember the isomers of glucose, alpha, the OH is below, alpha below, beta OH is above ABA. So we're looking at alpha glucose only today. So again, some questions here if you can answer. And again, I'm not going to necessarily go through these. You can look for the answers yourselves if you want. But let's have a quick look. So a lot of those questions we'll be answering in the next few moments, but we'll go through it anyway. So alpha glucose, basically alpha glucose polymers then, which are polysaccharides made up of long chains of alpha glucose repeating monomers. Are made up from repeated condensation reactions. So if you remember what we said the other video, a condensation reaction is where you remove water. So again, if I breathe on a window, I produce condensation on the window, and that means that I'm removing water from my body. So in a condensation reaction, you take out the water, which joins molecules up, and that causes the reactions to happen. And to make long chains of alpha glucose, you have repeated condensation reactions, and that changes the structure. So polysaccharides are different. So the two polysaccharides we'll be looking at is starch and then very briefly glycogen. So starch is the polysaccharide in humans, starch is the polysaccharide in plants, and glycogen is polysaccharide in animals that are used for energy storage. And both of those would be different from sugars because neither of them would be sweet, they wouldn't taste sweet, and more importantly for us, for storage molecules, they are insoluble in water. I'll go on to that in a moment. So this is what I was about to say. These are the 4 key characteristics of storage polysaccharides. So starch and glycogen both have these characteristics. And if you think about it, when we use respiration, when we do respiration, so in respiration, just to pause the video for a moment there, the glucose obviously needs to be used up. It's oxidized in respiration to produce energy in the form of ATP. Now that's absolutely critical when we get that glucose. So storage polysaccharides have to have the following characteristics. They are all compact, which means they can store a huge amount of glucose in a very small space to give you that energy store as and when you need it. They are in chains, and the reason they're in chains is that this helps. The long chains means you can hold lots of them in there, okay, and it means the glucose can easily be snipped off by an enzyme, so it can be grabbed hold of very quickly. When we look at storage polysaccharides, they are branched, and that gives it a larger surface area, which means the enzymes can get to the glucose molecules and release them from the storage polysaccharides really quickly. And the last one is they are insoluble. Now, insoluble is really important. So insoluble means it doesn't dissolve, and you might know that from GCSE for starch and for glycogen, because you needed to know they're insoluble then. But the reason that it's so important is that if it was soluble, if it was in a cell, okay, and we had such a high amount of glucose, so let's say all these glucose molecules were in the cell and that they weren't combined, and they were just individual glucose molecules, what that would do is it would lower the osmotic water potential, and as a result water would rush in to the cell due to osmosis happening from a high water potential to a low water potential, and that would cause a cell to burst. So it's absolutely vital that storage polysaccharides, which might contain thousands upon thousands of glucose molecules, are insoluble, because that means the sugar molecules don't interact with water because they're attached to each other, and therefore they have no osmotic effect. So these four characteristics are the same for glycogen and for starch. Now, in A-level biology, we actually look at starch more than glycogen. So it's important to understand the structure of starch. So if you look at this in the middle, this is a starch granule. And basically it's in layers of what we call amylose and amylopectin. So we're going to look at amylose first. Okay, now amylose is a long chain of alpha glucose molecules, and we're going to talk about that in a second. And then if you look at them on the top of this picture, amylopectin has branches coming off it, so very, very similar, but the difference is quite clear as well. So then you need to know about the structure of amylose. So amylose is basically a repeating chain of alpha glucose molecules. So if you look at each one of these alpha glucose molecules, it's a repeating chain, and because of the formation of the bonds, it causes a spiral structure to occur of amylase. And basically, if you're asked to describe what amylase is, you'd use your knowledge from earlier on in the course. So what you would say is it's a repeating chain of alpha glucose molecules. It might be over 1,000 alpha glucose molecules long. They have reacted together and joined together, forming a bond, forming a glycosidic bond. Okay, and the glycosidic bond is due to condensation reactions between the OH, or the hydroxyl group, of carbon 1 and carbon 4 of another atom. And they make up to 1,000 bonds, and they make a spiral shape as a result. Amylose is very, very, due to the spiral shape, it is very, very compact, and it is also insoluble to water. And a little bit extra for later on in the course, this explains the reason that iodine reacts to change colour in the food test with starch. So iodine, when it's as part of as a chemical in the iodine solution, comes in, okay, and the iodine is a yellowy brown colour. When the iodine reacts with starch, what it's actually doing is it's going in and it is reacting and getting caught effectively in the coils and then bonding with parts of the coil, and therefore that's why it changes colour, because it changes and it forms a bond with the starch molecule. So you got the second one. So we've done amylose, which is the long spiral in shape. Amylopectin is shorter than amylose, so it's not as long in a long chain. It is a spiral shape as well, which you can't see on here, obviously. So it's still sort of spiral in sort of structure, not really a spiral. But within that spiral shape, there are branches that emerge off the side of the amylose, off the side of the amylopectin. So amylopectin, very, very similar structure to amylose, but off the side of it, you get branches that form off the side. Now, what these branches do is they increase the surface area of the amylose, which gives you more chance for, sorry, I keep saying amylose, the amylopectin, which gives the enzymes a chance to break off those sugar molecules and allow them to be used for respiration. It's much more compact than amylose, so you have it. Now, you might also get sort of information about the actual bonding. So obviously the main structure of it is still carbon, a glycosidic bond between carbon 1 and carbon 4. So you've got carbon 1, carbon 4. Look at the more complicated picture at the bottom. Okay, most of your bonds in the chain are carbon 1 and carbon 4. Again, carbon 1, carbon 4, carbon 1, carbon 4, and carbon 1, carbon 4. But where the branches actually form, where the initial branches occur, it is carbon 1 and carbon 6. So if you look at this one here, Okay, this is your carbon 6 atom here, but this one here is carbon 1 of the branch. So what you have to have is a glycosidic bond between carbon 1 and carbon 6, or 1,6 glycosidic bond. And you see it being shown in a slightly different format here. So you've got carbon 6 here. After the bond's formed, you've got the glycosidic bond between that and carbon 1 of another atom. Okay, so that's just to sort of show you. So you don't need to be able to necessarily draw it, you just need to be able to describe it. It's the branching forms between carbon 1 and carbon 6 of another atom. So if we look at overall starch then, so you need to know, obviously you'll do much more about it being digested later on. GCSE, you need to know it's digested by the enzyme amylase into glucose. Alpha glucose, what we'll talk about now. Starch is a mixture of amylose and amylopectin. So it's about 30% amylose, 70% amylopectin. And then if we were to compare the two structures, basically of amylose and amylopectin, what we've just done in the last few minutes, amylose is the longer chain of about 1,000 alpha glucose molecules, and it is unbranched chain. And that unbranched chain is only made of 1,4 glycosidic bonds between the alpha glucose. Chains are coiled, but that is effectively that. The amylopectin then is different because it's shorter, so it might not be 1,000 alpha glucose molecules in length. Again, they are 1,4 alpha glucose chains, but where the branches form is 1,6 glycosidic bonds, which help form the branches, giving them a larger surface area, allowing the glucose molecules to come off. So the last bit is glycogen. So glycogen is like the animal starch, okay? So it's very, very similar in sort of structure. You have, again, identical alpha glucose molecules which join up to make a long chain which can be broken. Difference with glycogen to starch is the chains are a lot shorter, so you don't get like massive chains like amylose, and you've got more branches coming off. So lots and lots of branches between alpha glucose and one chain and alpha glucose and another chain. So you get this 1,6 glycosidic bond again there, and then you get branches that contain about 10 to 15 glucose molecules in a chain. Now, why that is so useful is because it means that the animals, which are obviously much more active and much more metabolically in need of energy, are able to have the glycogen has a larger surface area than starch does, so the enzymes are able to come along and say snip off that glucose molecule off there or that glucose molecule off there, and it means that we're able to have a higher level of metabolic activity, which is obviously essential for animals which are have much more energy demands as a result. Okay, so just finish off. There's some follow-up tasks if you want to do them. Please feel free to pause it. Other than that, I'll see you next time. Thank you very much.